Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Thank you for the opportunity for me to present our research with the title Screening of Simple Sequence Repeats Primers from Mutated Indigo Vera zolineriana plants. My name is Juatina Ida Royani. I am from BRIN. The background of our research: DNA profiling of plant using molecular marker is a useful tool for the identification and characterization of superior plants in plant breeding programs. This tool can shorten the time for superior plant selection, especially in plants with long growth cycles. Molecular marker work by revealing variation or polymorphism of the DNA sequences between different individuals in the population. One of the most popular marker is simple sequence repeats marker or microsatellite. Why? Because show codominance, highly polymorphic, hypervariable, and it is distributed throughout the genome and has a higher mutation rate. The molecular marker can detect rapidly the substitution mutation or point mutation, reassociation, insertion and deletion, replication error, and DNA tandem repeats. Mutated plants need to identification of genetic variation using a method that are simple, accurate, reproducible, and show the polymorphic between mutated plants to shorten time of the selection process. To use molecular marker with precision method, screening of primers that use is the main factor to obtain the optimal results. Selection of the optimal primer annealing temperature will increase the efficiency and specificity of amplification to obtain the suitable primer use. For this research, we use 14 SSS primers from same genus of Indigofera zolineriana that proved to amplify Indigofera pseudotinctoria. So the objective of the research was to screen 14 SSS primers to know the polymorphic DNA profiles of 12 mutated plants of Indigofera zolineriana. Material used for this research were leaves from mutated plants of Indigofera zolineriana with 6 different doses. Material preparation: mutated plants from Indigofera zolineriana with dose 0, 100, 200, 300, 400, and 500 gray were planted in the land. After 12 weeks planted, leaves were put into plastic clips and labeling in every sample for next treatment. For genomic DNA isolation, DNA genome from 12 mutated plants were isolated using Wizard Genomic DNA Purification Kit. For screening SSS primer, we use 14 SSS primers, 12 DNA genomes, and amplification with multiplex PCR with 2 PCR conditions. All product PCR then electrophoresis with 1 or 1.5% of gel agarose. For optimization of annealing temperature, the gradient temperature was set with 6 different temperatures based on the original temperature with the gradient degree at 0.3 degree Celsius. We used primer that amplified the DNA genome but had thin bands. So the result of genomic DNA isolation, 12 DNA genomes of mutated E. zolineriana leaves were successfully isolated using this protocol. Concentration of DNA genomes range between 345.6 until 3318.6 nanogram per microliter, and purity range between 1.56 until 1.90. For screening primer, screening with 40 SSS primers show that not of all primers which use could amplify the DNA genome of mutated Indigofera zolineriana in temperature of annealing at 59 degree, 5 primer show polymorphic, 8 monomorphic, and R4 primer couldn't amplify DNA genome. In temperature of annealing at 48 degree, 13 primers show polymorphic and R9 primer monomorphic. We assume that annealing temperature at 48 degrees show more polymorphic profile than at 59 degrees Celsius. DNA profiles of polymorphic bands show 1 until 10 bands, and the best primer was at 3 primer, with 1 until 10 bands with clearly bands, following by R8 primer with 1 until 8 bands, but with thin bands profiles. The thin bands show that temperature of annealing insufficient to attack the primer sequence to the DNA genome and still need to optimize the annealing temperature. For this screening, we obtain 5 primers that show polymorphic band at 59 degrees Celsius and 13 primers at 48 degrees Celsius. For optimization of gradient temperature, we only use R4, R5 into R5.2 DNA genome for treatments. The results show that every primer has specific TM for obtain clearly DNA band. After treatment for gradient temperature, the results show that 11 primers are suitable for use in further DNA profiling of another mutated Indigofera zolingeriana. The specific temperature that obtained from gradient temperature will be used as annealing temperature for the next PCR. So the conclusion of our research show that 12 DNA genomes of mutated Indigofera zolingeriana leaves were successfully isolated. 5 primers show polymorphic band at 59 degree and 13 primers at 48 degree. Treatment for gradient temperature, the results show that 11 primers are suitable for use in further DNA profiling. The specific temperature that obtained from gradient temperature will be used as annealing temperature for the next PCR. The authors would like to extend appreciation and thankful to Research Organization of Nuclear Technology, BRIN, for providing financial aid during this research. This is the part of our reference. So thank you for the attention. Assalamualaikum warahmatullahi wabarakatuh.